Hello friends, we will be discussing about the restrictive cardiomyopathy in this video. So basically what is cardiomyopathy? This cardiomyopathy means there is some abnormality in the heart muscle. So cardio means heart and myo means the muscle, and because this abnormality can be either the mechanical abnormality, that is there can be a defect in the contraction or dilatation, or there can be an electrical dysfunction in the conducting system, which again can affect the contraction and dilatation of the myocardial muscle. So that because of that abnormality, there can be inappropriate ventricular hypertrophy or the dilatation. So all these disorders which basically involve the abnormality in the heart muscle comes under the cardiomyopathy. So broadly it has been classified as primary cardiomyopathy and secondary cardiomyopathy. So primary cardiomyopathy means, as the name suggests, primarily it involves only the heart muscle, and secondary cardiomyopathy is a condition where we have a systemic disease which involves multiple organs, and in that one of the organ involved is the heart. So if it is secondary to certain systemic disorders, we call it as secondary cardiomyopathy. Now this is depending upon the etiology. Now, depending upon the clinical, functional and pathological patterns, we have three types of cardiomyopathies. That is dilated cardiomyopathy, hypertrophic cardiomyopathy and restrictive cardiomyopathy. Dilated cardiomyopathy, again as the name suggests, we have a dilated chambers of the heart, and heart is very enlarged and it is flappy, and we also have somewhat thinning of the ventricular walls. Whereas hypertrophic cardiomyopathy, again as name suggests, we have asymmetric hypertrophy of interventricular septum and the left ventricular wall. So this is the hypertrophic cardiomyopathy, and the restrictive cardiomyopathy is a condition where you have some infiltrative disorder which will be affecting the myocardium. Now the thickness of the myocardium will be same, it is not affected, but the function of the myocardium is affected. It cannot dilate. So there is a diastolic compliance in this. And among the three types of the cardiomyopathies, the most common one is the dilated cardiomyopathy, and the least frequent one is the restrictive cardiomyopathy. Now when we see the basic mechanism and the ejection fraction, in the dilated cardiomyopathy, the left ventricular ejection fraction will be less than 40%, and here the basic mechanism is we have a systolic dysfunction. Then we have a list of the causes, like it can be genetic cause or because of alcoholism, peripartum, myocarditis, hemochromatosis, chronic anemias, doxorubicin toxicity, Chagas disease, and it can be idiopathic also. Idiopathic is the most common cause in the dilated cardiomyopathy. Then the hypertrophic cardiomyopathy, we have the left ventricular ejection fraction will be 50 to 80%, and in the restrictive we have 45 to 90%. Both of them, the main mechanism of the heart failure is diastolic dysfunction. In the dilated it is the systolic dysfunction, whereas in the hypertrophic and restrictive it is the diastolic dysfunction. Then we have a list of causes for the hypertrophic: genetic causes, Friedreich's ataxia, storage disorders, and we can also say in the infants of diabetic mother. The causes I have dealt very detailedly in the individual videos of this. And the restrictive cardiomyopathy, the causes can be amyloidosis, radiation-induced fibrosis, idiopathic, and this we will see detailed in the next coming slides. You have infiltrative disorders, infectious diseases which can cause and all that. Now coming to the topic proper, this restrictive cardiomyopathy is a condition where you have decrease in the ventricular compliance. That means the dilatation of the ventricles doesn't occur properly, so that there is an impaired ventricular filling during the diastole. Now this leads to the decrease in the cardiac output, but here the contractile function of the left ventricle is usually unaffected. This is in the initial stages, but later on in some conditions even the contraction also is lost. So basically there is a decrease in the ventricular compliance, which leads to decrease in the ventricular filling. So the main mechanism is there is some infiltrative process, or there is a destruction of the myocardiocytes which causes the fibrosis. So we have a fibrotic process. because of which the dilatation of the myocardium is lost. So you have a reduced ventricular dilatation that leads to increased diastolic pressure and decreased diastolic volume because it cannot expand more. The pressure during the diastole is more and there is a reduced ventricular filling. Obviously that leads to decreased output, also cardiac output, and later on the patient goes into the cardiac failure. This is the basic mechanism in the restrictive cardiomyopathy. Now when we see the causes, they are basically divided into idiopathic causes or inflammatory causes, where we have endomyocardial fibrosis, Löffler's cardiomyopathy, endomyocardial fibroelastosis, and post-irradiation therapy. These all come under inflammatory causes. Then we have a storage disorders also, which can lead to the restrictive cardiomyopathy, that is glycogen storage disease and lysosomal storage disease like the Fabry's disease. And the infiltrative disorders like amyloidosis and sarcoidosis, we find restrictive. And then in the non-infiltrative conditions like hemochromatosis, which is a metabolic abnormality, and diabetic cardiomyopathy, systemic sclerosis, and some of the genetic causes. So all this can lead to restrictive cardiomyopathy. So basically all the causes we have divided in under 5 headings: idiopathic, inflammatory, storage, infiltrative, and non-infiltrative. So in the genetic causes, mainly it is caused by the mutations in the genes which express the sarcomeric proteins. If you remember tropomyosin C, I, beta myosin, heavy chain and all that. So mutations in these genes can cause restrictive. It depends upon the expression level also. If you have a high expression level, then you have increased production of the proteins, and they are more stable and they doesn't undergo the degradation. So you have accumulation of these proteins. So misfolded proteins, if you remember the mechanism of the misfolded protein, later on the accumulation of the misfolded protein leads to apoptosis of the cell. Similarly here you have the aggregation of this sarcomeric proteins, and later on this causes damage to the myocardiocyte, leading to the necrosis and later on fibrosis. So that causes the fibrosis and the stiffness. This is if you have increased expression of the genes responsible for the production of sarcomeric proteins. And if the expression level is low, obviously the sarcomeric proteins produced are less, then there is a contractile dysfunction that leads to impaired relaxation. So you don't have a proper sarcomeric proteins for the relaxations. So that also causes the diastolic dysfunction. So as a whole, if the expression is high or the low, both of them causes diastolic dysfunction only. Then the inflammatory causes, as I told you, we have 4 under this: endomyocardial fibrosis, Löffler's cardiomyopathy, endomyocardial fibroelastosis, and post-irradiation therapy. Now we'll see each one how they cause. When we see the endomyocardial fibrosis, this we see commonly in the children and the young adults, usually in the African countries and also in the tropical areas. Now the cause for this is some infectious diseases or the parasitic infections. So what they do is, when there is an infectious cause, there is activation of the inflammatory cells, and these inflammatory cells, they produce interleukins, they produce the growth factors also. So these interleukins, what they do is they stimulate the other inflammatory cells to produce the antibodies to destroy the infectious agent. So the antibodies which are produced here, they cross react with the myocardiocyte and then they cause damage to the myocardium. Then the growth factors also which are produced by these inflammatory cells, they stimulate the fibroblasts. So that causes the proliferation of the fibroblasts and you have deposition of the collagen. So here basically what happens is you have a damage to the myocardium and you have a deposition of the collagen. So this leads to the myocardial fibrosis. And if there is a parasitic infection, you have infiltration of the eosinophils. And eosinophils, they release the major basic protein that again causes damage to the myocardium. So whenever there is a damage to the myocardium and the myocardiocyte necrosis, later on the healing process is by the fibrosis. So fibrosis causes stiffness of the myocardium. There is a loss of elasticity. So that again leads to the diastolic impairment, causing the restrictive cardiomyopathy. This is how the restrictive cardiomyopathy occurs in endomyocardial fibrosis. Then another condition is Löffler's endomyocarditis. So here we have this condition. This is mainly because of increased eosinophils, hypereosinophilia, where you have eosinophilic infiltration into the myocardium. Now, increased eosinophilia or the eosinophils in the blood can be because of the two things. Either it can be primary eosinophilia or it can be secondary eosinophilia. Primary eosinophilia occurs in the chronic eosinophilic leukemias or hypereosinophilic syndrome, and secondary eosinophilia we see in the parasitic infections or some allergic conditions. So in both these conditions, whether it is primary or secondary, we have increase in the eosinophil count, and these peripheral eosinophilia, in this condition we have infiltration of the eosinophils into the different organs in the body, including the heart. So when they enter into the heart, the major basic proteins which are produced by the eosinophils, they cause damage to the myocardium, and there is a myocardiocyte necrosis that leads to fibrosis again as a healing process. And not only that, when the contraction is reduced and when there is a damage to the myocardium, that can also lead to the thrombus formation and further complications for this. Another one is endocardial fibroelastosis. This is a very rare condition. We see the fibroelastic thickening that typically involves the left ventricular endocardium, and this usually we see in the first 2 years of the life, and the cause is thought to be either because of some viral infections, typically in the intrauterine exposure to mumps, or it is because of the mutations in the gene for the tafazin. So these two are the main proposed causes for this, and typically it occurs in the first 2 years of the life, and it also involves aortic valve, producing aortic valve obstruction, and the patients also will have other congenital cardiac anomalies. So if the diffuse involvement occurs, patient goes into the cardiac failure and you have cardiac decompensation and the death of the patient. So in this condition, the fibrosis, it starts from the apex and goes towards the tricuspid or the mitral valve, and here the fibrosis is typically in the endocardium or the subendocardium only. So the how the fibrosis occurs is it's from the apex towards the valvular region, and when the fibrosis occurs, the volume of these chambers and the compliance of this ventricular chambers is reduced, so you have defect in the ventricular filling. And also because the contraction is reduced in the elastosis, there can be the stasis, which can cause the ventricular mural thrombi formation. Now, how does the radiation cause? So whenever there is a radiation-induced injury, radiation can cause increased production of the oxygen-free radicals. So there is a release of the reactive oxygen species which cause damage to the myocardium. So obviously there will be again the necrosis, and the healing process is by the fibrosis. So fibrosis will again cause the restrictive cardiomyopathy. Now in the infiltrative we have amyloidosis. I think everybody knows amyloidosis is a condition where we have deposition of the fibrillary protein which has a beta pleated sheet structure or the configuration. So it is insoluble, and where in this condition we have deposition of all this fibrillary protein. So cardiac amyloidosis, it occurs as a part of the systemic amyloidosis, or it occurs in the just senile cardiac amyloidosis where mainly the heart is involved. As a part of systemic amyloidosis, we see in a condition of the plasma cell tumors where we have increased production of the light chains. So there is an amyloid light chain deposition in the multiple organs along with the heart. And another condition is in the severe chronic inflammations where you have increased production of the interleukins by the inflammatory cells, which stimulates the liver, and the liver produces serum amyloid associated protein. Now this is deposited in multiple organs along with the heart. So here the heart occurs, the deposition of amyloid in the heart occurs as a part of the systemic process. Whereas another condition where we just have senile cardiac amyloidosis where only mainly the heart is involved. So when we see normal condition, the liver produces this transthyretin and the transthyretin molecule is a tetramer. So in certain conditions where you have a genetic abnormality, monomers are produced, and these monomers of the transthyretin, they are very resistant to the degradation. They are insoluble. So these transthyretin molecules gets deposited in the heart, and that is the cause for the senile cardiac amyloidosis. So in the amyloidosis, heart is involved as a part of senile cardiac amyloidosis or as a part of secondary amyloidosis. Now here in the amyloidosis, how the heart will be, the gross morphology is the heart will be firm and rubbery because of amyloid deposition, and you have the thickened walls because of the deposition. The wall thickness increases, and the cut section there will be the small translucent nodules which will appear waxy, and this usually we see on the atrial endocardial surface. Okay, so we have thickened walls and we have a waxy surface. Cut section, it is semi-translucent and waxy in appearance, and you have those nodules of the amyloid deposition. And when we see microscopically, we see the eosinophilic amyloid deposition in between the myocardiocytes, or along with that we can see in the conduction tissue valves in the endocardium, pericardium, and also in the intramural coronary arteries. In all this we can see the deposition of this amyloid material. So when it occurs in the wall of the vessels, what does it do is it reduces the lumen of the vessel. So there can be ischemia to the myocardium, that again further causes damage to the myocardium. Now another condition is a sarcoidosis. Sarcoidosis is a granulomatous disorder, non-caseating granulomas we see in this, and cardiac involvement in this is very rare. It usually constitutes in the 10% of the cases only. It involves all the three, endocardium, myocardium and pericardium, but most commonly it involves the myocardium. We see granulomas in the myocardium, and it also involves the base of the interventricular septum. So when it involves the interventricular septum, obviously the conduction system is also affected in this. So when the granulomas are formed, they destroy the myocardium and there will be the fibrosis, which causes the restrictive cardiomyopathy. So mainly the left ventricle and the conduction system is affected. The right ventricle is indirectly affected by this. You have a back pressure in the right ventricle, and there can be the pulmonary fibrosis, pulmonary hypertension, and left ventricle, or there can, because of the left ventricular dysfunction. Now another condition is hemochromatosis, where you have increased deposition of the iron in different organs. So if you remember the cell injury chapter, the freely available iron ions, they help in the production of the reactive oxygen species via the Fenton's reaction. So similarly in the myocardiocyte, when you have increased deposition of the iron, that iron is utilized for the formation of the free radicals. Now these free radicals, they cause the oxidative stress. So when there is an oxidative stress in the myocardiocyte, there is an impaired coupling of excitation contraction. You have impaired contraction relaxation of the myocardium, elevated cytoplasmic calcium levels, and the free radicals, they cause lipid peroxidation of the membrane, damage to the DNA, and even they cause apoptosis. So apoptosis induced by the iron, we call it as ferroptosis. So these all the changes occur when there is an increased reactive oxygen species, which are produced by the Fenton's reaction in the availability, when there is an increased availability of iron ions. Not only this, iron also stimulates the fibroblasts. So when there is an activation of the fibroblasts, they proliferate and they cause an increased collagen formation. So that causes the fibrosis. So ultimately, because of this or this, there is a restrictive cardiomyopathy. Now the storage disorders, if you see, we have glycogen storage disorders and we have a lysosomal storage disorders. So Fabry's disease is one of it. This is caused by deficiency of alpha galactosidase A lysosomal enzyme. Now when there is a deficiency of this enzyme, we have the accumulation of globotriaosylceramide, GB3, in the lysosomes of the cardiomyocytes. So not only in the cardiomyocytes, we see in the valvular fibroblasts and also in the conduction system. So when we have a deposition of the substrate in a cell, the size of the cell increases. The size of the cell increases, the size of the entire organ also increases. So we have left ventricular hypertrophy and you have a papillary muscle hypertrophy also. So later on you have a damage to the cell itself. So there will be a myocardiocyte damage, necrosis and fibrosis. So finally there will be a diastolic dysfunction with the restrictive cardiomyopathy. Similarly, it occurs in the glycogen storage disease also. In the glycogen storage disease, there can be abnormality in the enzymes. There can be deficiency in the enzyme, or there can be a functional defect in the enzyme, or there can be a disturbed molecular transport. Any of it, it causes the accumulation of the substrate. Similar like what we have seen in the Fabry's disease, accumulation of the substrate causes the hypertrophy. First initially it will be the hypertrophy, later on it will be the damage to the myocardiocyte leading to the cardiomyopathy and valvular dysfunction. And when we see in the diabetes is a condition where we have hyperglycemia, hyperlipidemia, lipotoxicity will be there, increased advanced glycosylation end products and increased angiotensin 2. So all these things, they cause damage to the cells. How they cause is, they cause some metabolic defects. So here in the myocardiocytes, we have metabolic changes because of the mitochondrial dysfunction. So because of the mitochondrial dysfunction, the first thing what happens is there will not be the recruitment of the glucose transporter type 4 molecules on the membrane. So if these transporters are not recruited on the membrane, glucose cannot enter into the cell. So there is a decrease in the intake of the glucose, and along with that there is a lowered calcium pump activity in the sarcoplasmic reticulum. Because of this, there is an increased calcium levels in the cytoplasm causing damage to the cardiomyocyte. Not only that, this also causes the oxidative stress, which can directly cause damage to the cardiomyocyte, causing the death of them and later on fibrosis, or they can also produce the inflammatory reaction, and inflammatory reaction produces, as I told you, we have the growth factors also which are produced. So we have a pre-fibrotic response and a cardiac fibrosis. It can also damage the endothelial cells, and these endothelial cells will cause microvascular dysfunction, because of which there can be ischemia to the myocardium. So that also causes the fibrosis and cardiac dysfunction, or otherwise because of the mitochondrial dysfunction there is decreased insulin stimulated coronary endothelial nitric oxide synthase activity. So if there is a decreased nitric oxide synthase activity, there is decreased nitric oxide production. So when there is a decrease in the nitric oxide production, there is a phosphorylation of the titin molecule, and this phosphorylation of the titin causes stiffening of the titin, which decreases the compliance of the cardiomyocyte. It cannot dilate cardiomyocyte. So because of the stiffness, so there is a restrictive cardiomyopathy. Again, we have the cardiac dysfunction. And another condition is systemic sclerosis. Here we don't know what is the cause. There is some unknown stimuli which stimulates the antigen presenting cells, and these antigen presenting cells, they indirectly stimulate the CD4 plus T cells, and these CD4 plus T cells, when they are stimulated, they produce the growth factors and they also produce the cytokines. Both of them, they stimulate the fibroblastic activity, and you have proliferation of the fibroblasts and deposition of the collagen. So typically in the myocardium, we have microvascular abnormalities, like because of the concentric intimal hypertrophy of the small coronary arteries leading to the ischemia, and you have later on you have cardiomyocyte damage and the myocardial fibrosis. Because of this microvascular abnormality, the blood supply is reduced, which causes damage to the cardiomyocytes. And systemically in different organs, or typically in pulmonary vessels, there is a thickened intima, narrowed lumen, which leads to ischemia in the different organs. So that is the cause of restrictive cardiomyopathy in the systemic sclerosis. Now, as a whole, when we see the morphology of the heart in the restrictive cardiomyopathy, we have the fibrosis of the subendocardial or the endocardial fibrosis of the left ventricle or the other ventricle, which causes biatrial enlargement. Because of the back pressure, we have the biatrial enlargement, but the ventricles can be of normal size or sometimes they are reduced in the size also. And there is diffuse or patchy interstitial fibrosis. When we see microscopically, we see the fibrosis. So main change what we see is biatrial enlargement. Because the ventricles cannot dilate, the atrium will have the back pressure. So both the atria will be dilated. So biatrial enlargement will be present. And we have the ventricles which are of the normal size or reduced size. And microscopically, we see the fibrosis. And what can be the treatment is we have to treat the cause, like if it is an inflammatory cause or if it is a diabetes or infiltrative causes. So or the causes, if we treat, we can reduce this, or otherwise we have to go for the heart transplantation. Now that finishes the restrictive cardiomyopathy. So if we see the summary, first we should know what is the definition. Definition is we have a disorder of the heart muscle where you have decreased compliance resulting in the impaired ventricular filling. Now the causes, we have divided them into under the five categories: idiopathic, inflammatory, where we have endomyocardial fibrosis because of some inflammatory reaction. Löffler's cardiomyopathy, this is because of increased eosinophilia, primary or secondary, which causes damage because of the major basic protein. Endomyocardial fibroelastosis, which we see in the 2 years age, and usually it's a rare condition, and this is because of the titin gene and post irradiation therapy, which causes the fibrosis. Endomyocardial fibroelastosis because of tafazine gene, mutations in the tafazine gene, or it is because of the viral intrauterine infections caused by the mumps. That has been thought to be the cause. And post-irradiation therapy, damage to the myocardiocyte because of the oxidative stress that causes the fibrosis. And in the storage disorders, we have glycogen storage disorders and the Fabry's disease, where we have accumulation of the substrate in the cardiomyocytes or in the myocardium, causing damage to them. Then in the infiltrative disorders, we have amyloidosis and the sarcoidosis. Then the non-infiltrative, we have hemochromatosis, diabetic cardiomyopathy, systemic sclerosis, and the familial or the genetic causes. So all these are the causes. And then when we see the morphology, as I told you, because of the fibrosis, there is a fibrosis which typically involves endocardium or the subendocardium, later on the myocardium also. So this causes diminished compliance. Because of this, there is a back pressure in the atria. So we have biatrial dilatation, and initially we have the ventricles of the normal size. Later we can have the diminished volume of the ventricles also. So treatment part, when you see, either you have to do the treatment of the underlying cause. If not possible, we have to go for the heart transplantation. So you have to remember the definition, the causes, morphology, and the treatment. So that finishes the restrictive cardiomyopathy. Thank you, friends. Thank you for listening patiently.